Good morning, everyone, and thank you for joining us today. I'm Dr. S.K. Singh, and I'm excited to share insights on sterility assurance through 0.2 micron filtration. Today, we'll explore the key challenges, limitations, and the importance of compliance in ensuring effective sterilization. Over the course of this presentation, I'll guide you through the critical aspects of filtration processes, highlight potential hurdles, and discuss best practices to meet industry standards. Let's get started. Welcome to our discussion on sterile filtration using 0.2 micron membrane filters. This process is absolutely vital in pharmaceutical manufacturing, especially for heat-sensitive products that can't undergo terminal sterilization. The main goal here is to remove microorganisms and particulate contamination, ensuring that the product remains pure and effective. At the same time, we need to preserve the product's quality and integrity throughout the process. It's important to note that regulatory authorities worldwide recognize 0.2 micron filters as sterilizing-grade filters, but only when they are properly validated and controlled. Now, let's talk about why this is so crucial. First, it guarantees the sterility assurance of the final product. Second, it helps prevent microbial contamination, which could otherwise compromise safety. This filtration supports aseptic manufacturing operations, which are essential for maintaining a sterile environment. It ultimately protects patient safety by ensuring that only safe, uncontaminated products reach the market. And of course, adhering to these filtration standards also helps us stay compliant with good manufacturing practices, GMP, which are the backbone of quality in our industry. So, in essence, sterile filtration with 0.2 micron filters is a cornerstone of producing safe, effective, and high-quality pharmaceutical products. Let's take a closer look at the applicable regulatory guidelines that shape our sterilization and aseptic processes. First, we have key references like the U.S. FDA regulations, specifically 21 CFR Parts 210 and 211, which set the foundation for pharmaceutical manufacturing practices. In Europe, the GMP Annex 1 provides detailed guidance on sterile medicinal products, emphasizing the importance of maintaining sterility throughout production. The USB 1229 focuses on sterilizing filtration, highlighting best practices for sterilization through filtration methods. PDA Technical Report Number 26 offers practical insights into sterilizing filtration processes, while ISO 13408 provides standards for aseptic processing, ensuring consistent quality and safety. World Health Organization GMP guidelines and PIC-S sterile manufacturing guidance further reinforce the global expectations for sterile manufacturing, promoting harmonization across regions. Lastly, ICH Q9 emphasizes the importance of quality risk management, guiding us to identify, evaluate, and control risks effectively. Moving on to regulatory expectations, it's critical that we use validated sterilizing-grade filters to ensure sterilization efficacy. Integrity testing before and after filtration is essential to confirm that filters are functioning properly and maintaining sterility. A robust contamination control strategy, or CCS, is fundamental. This involves comprehensive measures to prevent contamination at every stage. Effective bioburden management helps us monitor and control microbial load throughout production. And of course, complete documentation and traceability are vital. Keeping detailed records ensures accountability, facilitates audits, and maintains compliance with regulatory standards. All these elements together help us meet regulatory expectations and uphold the highest quality standards in sterile manufacturing. Let's take a closer look at the scientific basis of the 0.2 micron filtration. First, the principle of sterilizing filtration is primarily based on size exclusion. Essentially, the filter acts like a sieve, trapping particles and microorganisms that are larger than 0.2 microns. But it's not just about size. Adsorption also plays a role, where contaminants stick to the surface of the filter. Additionally, the membrane pathways are tortuous, meaning they have a complex winding structure that makes it even harder for microbes to pass through. Now, when it comes to validation, we typically use Brevandemonas diminuta as our standard organism. This bacterium is particularly small and robust, making it the perfect challenge organism for testing filter effectiveness. The scientific justification for using a 0.2 micron filter hinges on the fact that most pharmaceutical bacterial contaminants are larger than this size. So, when the filter is properly validated, it reliably retains these microbes, ensuring safety and sterility. Our main objective here is to demonstrate that this filtration method can consistently retain microbes, even under worst-case processing conditions. This gives us confidence that the filtration process is both reliable and effective, which is crucial for maintaining product safety in our industry. Let's take a closer look at the role of 0.2 micron filters in sterility assurance. These filters are essential in various pharmaceutical applications, ensuring that products are safe and free from microbial contamination. For example, they are widely used for injectable products, where sterility is absolutely critical, as well as for ophthalmic preparations, vaccines, biologics, sterile APIs, and even media and buffer filtration. One of the key benefits of these filters is their effective bacterial retention. They act as a reliable barrier, preventing bacteria from passing through. Plus, they are especially suitable for heat-sensitive products, which cannot withstand traditional sterilization methods. This makes them invaluable in aseptic processing, helping maintain the integrity of delicate formulations. Another important point is that they help preserve product stability and potency, ensuring that the final product remains effective until it's used. Now, speaking about regulatory acceptance, these 0.2 micron filters are recognized worldwide as sterilizing-grade filters, provided they are properly validated. This global recognition underscores their importance and reliability in the pharmaceutical industry. Overall, integrating these filters into your processes supports both compliance and product quality, reinforcing the overall theme of ensuring patient safety through robust sterilization methods. Let's take a closer look at the operational impact of challenges in sterile filtration. One of the key issues we face is reduced filtration efficiency, which can slow down our overall process and affect product quality. Batch processing delays are another concern, as any interruption can cause significant setbacks in production timelines. These delays also increase the risk of sterility issues, which is critical in maintaining product safety and compliance. Moving on to the specific challenges in sterile filtration, we often encounter filter fouling and blockage. These problems reduce flow rates and can even cause the system to shut down unexpectedly. A high bioburden load complicates filtration further, making it harder to achieve the desired sterility. Additionally, product adsorption to the membrane can lead to product loss and variability, affecting overall yield. Flow rate reduction is a common symptom of these issues, often accompanied by pressure fluctuations that indicate clogging or membrane stress. Compatibility problems between filters and process fluids can cause material degradation or leaks, which compromise integrity. Lastly, incomplete wetting of the membrane can impede filtration performance right from the start, leading to inconsistent results. All these challenges underscore the importance of selecting the right filtration systems and maintaining rigorous process controls. Addressing these issues not only improves operational efficiency, but also ensures the safety and quality of our sterile products. Let's talk about some of the limitations we face with 0.2 micron filtration, both from a scientific and process perspective. First, it's important to note that this filtration method is not effective against certain viruses and endotoxins, which can sometimes bypass the filter. Additionally, the retention of mycoplasma can vary depending on specific conditions, so it's not always completely reliable. Another key point is that 0.2 micron filters do not remove dissolved contaminants, meaning some impurities can still pass through. There's also the risk of product loss due to adsorption, where some of our product might stick to the filter material itself. And finally, membrane damage can occur, which could compromise the sterility assurance we rely on. From a regulatory standpoint, it's crucial to understand that filtration alone isn't enough. Proper contamination control measures need to be in place to ensure safety and compliance. The main objective here is to understand these process limitations thoroughly. By doing so, we can apply the right controls and strategies to mitigate risks effectively and ensure the integrity of our products. Let's talk about managing prefiltration risks and why bioburden control is so important. When there's an excessive microbial load in your material, it can cause several issues. For example, it can overload the filter, making it harder for it to do its job effectively. It can also increase the filtration pressure, which may lead to filter failure or damage. And most critically, a high microbial load can reduce the filter's ability to retain microbes, potentially allowing contaminants to pass through. So, what measures can we take to control these risks? First, maintaining a controlled manufacturing environment is essential. This means proper cleanroom standards and environmental controls. Timely processing of materials is also crucial. Delays can lead to microbial growth. Ensuring all equipment and transfer lines are properly sanitized helps prevent contamination. Using effective prefiltration systems can reduce bioburden before the main filtration step. And implementing robust environmental monitoring programs allows us to detect and address any contamination issues early. Now, from a regulatory standpoint, the focus is increasingly on integrating filtration into the overall contamination control strategy, or CCS. This means filtration isn't just a standalone step, but a critical part of a comprehensive approach to maintaining product purity and safety. By doing so, we ensure that our processes are aligned with regulatory expectations and that we are proactively managing contamination risks throughout manufacturing. Now, let's talk about the validation of sterilizing filters. When we validate these filters, we need to conduct several key studies to ensure they perform effectively and safely. First, bacterial retention testing is crucial. It confirms that the filters can effectively remove bacteria from the fluid. Next, we look at product compatibility studies to make sure the filters won't adversely affect the product itself. We also evaluate extractables and leachables, which helps us understand if any unwanted substances could migrate from the filter into the product. Adsorption assessments are important too, as they reveal whether the filter might adsorb or remove active ingredients from the product. Flow rate and pressure studies are conducted to verify that the filters allow the required flow without causing excessive pressure, ensuring efficient processing. And finally, we perform integrity test correlation to confirm that integrity tests accurately reflect the filter's ability to retain contaminants. From a regulatory standpoint, the key expectation is that validation must demonstrate that the filters consistently produce sterile filtrate every time, without compromising the quality of the final product. This means all these studies come together to provide a comprehensive picture of filter performance, giving confidence that the sterilization process is both effective and reliable within our overall quality framework. Now, let's talk about ensuring filter performance, which is a critical part of maintaining quality and safety in our processes. First, we'll look at some common integrity tests used to verify filters are functioning properly. These include the bubble point test, the diffusion test, and the pressure hold test. Each of these tests helps us confirm that the filter is intact and capable of performing its intended function without leakage or failure. Moving on, there are regulatory expectations we need to be aware of. Specifically, under EU GMP Annex 1, pre-use and post-use integrity testing, often called PUPSIT, are emphasized. The key point here is that integrity testing must not only confirm the physical integrity of the filter, but also correlate with its ability to retain microbes. This ensures that the filter is truly effective in preventing contamination. The main objective of these integrity tests is to verify the filter's integrity both before starting filtration and after completing the process. This way, we can be confident that the filtration has been successful and that the final product remains safe and uncontaminated. Overall, these measures are vital to maintaining compliance and ensuring product quality throughout the process. Let's take a closer look at the regulatory expectations outlined here. First, it's essential to recognize that applying ICH Q9 quality risk management principles is fundamental. These principles guide us in systematically identifying, evaluating, and controlling risks throughout the product lifecycle. In terms of compliance strategies, implementing risk-based contamination control measures is key. For example, incorporating redundant filtration where appropriate adds an extra layer of safety, ensuring that if one filter fails, another can take over. Setting up defined alarm and monitoring systems helps us stay alert to any deviations in real time, allowing for prompt action. Preventive maintenance programs also play a critical role. They help us maintain equipment performance and prevent contamination before issues arise. Now, when we talk about a quality risk management approach, it's about making informed decisions based on key risk assessment factors. These include product characteristics, which influence how we approach filtration, filter compatibility, ensuring materials are suitable for the product, and bioburden levels, which tell us about microbial contamination risks. Other factors like filtration duration and pressure are vital because they affect the effectiveness and integrity of the process. Environmental conditions, such as temperature and humidity, can impact contamination risks as well. And finally, operator interventions. How and when operators interact with the system must be carefully considered to minimize human error. All these elements are interconnected, and understanding them helps us develop a robust contamination control strategy that aligns with regulatory expectations and ensures the quality of our products. Let's take a closer look at some common regulatory observations that we often see. These are the deficiencies that regulators frequently identify during inspections. First, inadequate filter validation is a key issue. This means that the filters used in production haven't been properly tested to ensure they perform as expected, which can compromise product quality. Next, missing integrity testing records are a concern, because they suggest a lack of proper documentation and verification of filter performance. Poor bioburden control is another critical point. If contamination levels aren't properly managed, it can lead to compromised sterility. In addition, inadequate change control procedures can pose risks because changes to processes or equipment aren't being controlled or documented properly. We also see incomplete investigations of filtration failures, which means issues aren't thoroughly understood or resolved. Lastly, a weak contamination control strategy can leave the entire process vulnerable to contamination risks. Now, what are the potential consequences of these deficiencies? They can lead to serious regulatory actions, such as FDA warning letters, which can harm a company's reputation and operational standing. Product recalls are another consequence, leading to financial losses and patient safety concerns. Sterility assurance failures are particularly alarming because they directly impact patient safety. Regulatory bodies also record inspection observations and may issue import alerts, which can restrict product distribution and market access. Overall, these common deficiencies highlight the importance of robust validation, documentation, and contamination control measures to maintain compliance and ensure product safety. Now, let's take a look at some of the emerging technologies and improvements that are transforming sterile filtration systems. One major development is the rise of single-use sterile filtration systems. These are incredibly beneficial because they minimize cross-contamination and reduce cleaning validation times, making processes more flexible and quicker to implement. Another exciting advancement is the use of automated integrity testing equipment. This automation ensures that filters are functioning properly without manual checks, saving time and increasing reliability. Coupled with real-time pressure and flow monitoring, these systems provide continuous data, allowing us to detect potential issues early and maintain optimal filtration performance. AI-assisted filtration monitoring is also making its mark. By leveraging artificial intelligence, we can analyze vast amounts of data to predict filter failures or performance deviations before they happen, which significantly enhances process reliability. Finally, the development of advanced low-protein binding membranes helps reduce product loss and contamination, especially important for sensitive biopharmaceuticals. All of these innovations collectively lead to several benefits. They improve process reliability, reduce the risk of contamination, boost operational efficiency, and strengthen traceability and data integrity. These improvements align perfectly with our goal of ensuring safer, more efficient manufacturing processes. Let's now look at some best practices for sterility assurance, particularly when it comes to strengthening filtration compliance. First, it's crucial to always use validated sterilizing-grade filters. These are specifically designed and tested to reliably remove microorganisms, which is fundamental to maintaining sterility. Maintaining effective bioburden control is equally important. This means keeping microbial levels in check throughout the process. Routine integrity testing of filters helps ensure they haven't been compromised, and periodic revalidation is necessary to confirm that our sterilization processes remain effective over time. Implementing a robust contamination control strategy, or CCS, provides a comprehensive approach to preventing contamination. Training personnel thoroughly in aseptic filtration techniques is vital. People are often the first line of defense. Complete documentation and traceability are key. They allow us to track everything and quickly identify any issues that arise. Additionally, trending filtration deviations and failures gives us insight into potential weaknesses, so we can proactively address them. Now, moving into continuous improvement, it's all about refining our processes. Implementing CAPA, or corrective and preventive actions, helps us systematically address issues and prevent recurrence. Regular risk reviews and process optimization ensure we're always aligned with the best practices. And of course, monitoring emerging regulatory expectations keeps us compliant and ahead of industry changes. All these measures together form a strong foundation for ensuring sterility and product quality throughout our operations. Let's wrap up by emphasizing that 0.2 micron filtration is absolutely a critical component of ensuring sterility in pharmaceutical manufacturing. This small pore size plays a big role in protecting patients and maintaining product quality. Scientific validation and integrity testing are essential here. They provide the evidence and confidence that our filtration processes reliably retain microbes, which is fundamental for sterility assurance. However, it's important to recognize that filtration does have its operational and scientific limitations. No process is perfect, so robust risk management strategies are necessary to address these challenges and prevent contamination risks. Effective contamination control and lifecycle management are also key. They not only help us stay compliant with good manufacturing practices, but also ensure that our filtration systems remain effective over time. And finally, it's exciting to see how advanced filtration technologies are continually evolving. These innovations are improving not only the level of sterility assurance, but also enhancing operational performance, making our processes more efficient and reliable. All of these elements come together to reinforce the overall theme, that maintaining sterility is a comprehensive effort, combining proven science, risk management, and technological advancement. For further support, please feel free to reach out to us. We're here to help you with any questions or additional resources you might need. And with that, I want to sincerely thank you all for your time and attention today. It's been a pleasure sharing this information with you, and I hope you found it valuable. Remember, your success is our priority, so don't hesitate to get in touch if you need any assistance. Thank you once again.